So I'm going to try to squeeze two things into the next 50 minutes or so. We're going to give a very brief overview of some of the concepts behind the modeling, sort of the physical, chemical underpinnings of the kinds of modeling that we do in virtual cell and COPACI. So I'll be talking about reaction kinetics and diffusion, but on a very, sort of overview level, hopefully giving you enough information so you can begin to think about your biological systems in quantitative terms. And then the nice thing is that both COPACI and VCell, virtual cell VCell, will then allow you to build your models with these sort of minimal this sort of minimal background. As long as you kind of understand what reaction kinetics and diffusion are about, which hopefully you all had as undergraduates in your chemistry courses, you'll be able to model with these tools. They're designed to be used by people who are not physical chemists, who are not physicists, who are not computer scientists or mathematicians. They're designed to be used by biologists, but biologists who want to be quantitative, who want to think quantitatively about their projects. So there are more complete lectures online. There's a full lecture on reaction diffusion. That's a full hour long that you can get more information about. And we deliberately don't go into that detail because it's online. And likewise, there's a full lecture on virtual cell that's also available, a full hour lecture. The reaction diffusion lecture is by myself, and this lecture on VCell is by Ann Cowan. All right, so without further ado, let's get to the reaction kinetics. So, again, hopefully you kind of can resurrect this from your undergraduate days, but you remember that if you have a molecule reaction of A going to B, it usually has to get over an energy barrier. And the size of that energy barrier before it can transition from A to B is called the transition energy. And the higher that barrier is, the slower the reaction is. So let's just say that there's enough thermal energy to get over that barrier, but with a 10% chance every second. So if you had 10 molecules of A at time 0, how many would there be at 1 second? So you can all think about that for a second. I don't want you to necessarily give me the answer. I'll give it to you. The answer is that we really actually don't know how many B molecules there will be, because this is a probabilistic event. Chances are good that if there are 10 A molecules and there's a probability of one jumping the barrier every second, that at least one, maybe several, will have jumped the barrier and be converted to B. But we don't know for sure. And so we have to we have to think about this as a stochastic process, as a probabilistic process. So for a small number of A molecules, like 10, this would be stochastic. And we have to sort of roll the dice each time. And when we do a simulation, we would do a stochastic simulation, which assigns that probability and follows the reaction in terms of a trajectory that could be different every time we run the simulation. And I'll show you that in a second. But if there are a large number of molecules, and we know that the probability is 0.1 per second, for example, then we can solve that deterministically. We can write down a differential equation. And the differential equation for this case would be that the change in the concentration now of A, not in the number of molecules of A, but the number of molecules per volume, the concentration, would be equal to minus 0.1, because we're losing one with a probability of 0.1 per second, times the concentration of A. And you can solve that differential equation, as hopefully all you know from your math, and it turns out to be an exponential decay, where the starting point is the concentration of A at time 0. So hopefully, again, all of you are familiar, and I'm just sort of reminding you of how this works. But as I say, the good thing is that if you can express some of these reactions quantitatively in a model, the software will actually calculate this deterministic expression for you. We'll solve the integral, solve the differential equation, we'll integrate the differential equation. So here's an example of one of these runs where we have a stochastic probability of 0.1 per second. And you can see that there's, sometimes it takes very little time for a transition to happen, 10 molecules going to 9. Sometimes it takes a little longer, but eventually you get down to very few A molecules after 20 seconds here, and correspondingly more B molecules. And if you solve the differential equation, the deterministic continuous equation, then this is an exponential decay of A and an exponential rise of B. Okay? So far, no hands up. So hopefully that means everybody is on the same page. An important point, of course, is that if you have many A molecules to begin with, instead of just 10, let's say you have 1,000, then the curves still look a little noisy for a single trajectory, as you can see here, but they start to look a lot more like the deterministic solution. And if you run the stochastic simulation 1,000,000 times, you wouldn't be able to see any of this noise anymore. It would be a very smooth curve. The average of 1,000,000 of these runs would be very smooth, and it would look exactly like the deterministic solution. Okay. So as I said, the software called PASI or Virtual Cell can solve these kinds of equations for you. And for a simple case like this, you could solve it with pencil and paper. You don't need the software if you know a little bit of the math. But even if you know that math, for a more complicated reaction scheme, and this is just a series of complicated pathways where each of the green balls represents a molecule or a state of a molecule, and each of the yellow squares represents a reaction that combines those molecules. And for something as complicated as this, there's no way to solve it with pencil and paper, even if your math is really good. And so you absolutely need the software to be able to solve these kinds of kinetic systems. And here's an example of a solution where I've highlighted some of the variables, and you can see how they change as a function of time. It looks like there was some sort of stimulus at about 100 seconds that set off some of these other changes. Okay. So you can we're going to teach you how to use both virtual cell and COPASI to solve problems like this. There's another kind of problem. Instead of solving reaction kinetics, you might also be interested in what's called the steady state. What happens after everything settles down? What is the behavior of the system when the rates of the reaction are all equal to 0? So that doesn't mean, I mean, obviously one solution for all of the rates being equal to 0 if all of the concentrations are 0. That's not the solution that we're interested in. We're interested in all of the situation where all of the concentrations are balanced so that they do exist. There is a steady concentration of A, B, and C, and D in this case, in this little reaction diagram. But they don't change because the rates, which in this case for the second rate would be the concentration of C times the rate constant minus the concentration of D times the reverse rate constant, where all of those rates are equal to 0. And that's called the steady state. All species have constant concentrations. To calculate the steady state concentrations, all you need are these equations, these equations here showing the relationship of the rate constants. The concentrations are all equal to 0, as well as the mass conservation relationships. So you know that if A plus B goes to C and C goes to D, that there's some relationship between the initial concentrations of A and B and how much C you could possibly get. You can never get more C than you have either A or B, right? And so those mass conservation relationships are also algebraic relationships, which can produce a set of simultaneous algebraic equations that can be solved. And again, you could do a simple case like this with pencil and paper, just solving the algebra. But if you have a more complex system, like the one I showed before, then you need a software tool like COPASI. And COPASI can solve for the steady state automatically, and it's a very good use of that particular tool. You can use either COPASI or virtual cell to solve the steady state in another direct way. You can just start a kinetic simulation using any combination of initial conditions and let it run out to a very long time. Let it run out to where you're sure things aren't changing anymore. And that can also produce a steady state. So that's sort of like a very quick and dirty way of figuring out what the steady state might be. There are cases, which I'm sure Pedro will talk about, where you can have oscillations that are actually a steady state solution. And for those, you might want to use more sophisticated methods. But one important point that I want to make in terms of modeling your systems, very often, especially in cell signaling, you want to apply a stimulus. Let's say you want to produce a response to a ligand binding to a receptor. So at time 0, you'll add some concentration of that ligand. It will start binding the receptor, and things will start happening downstream. The downstream events may involve phosphorylation, calcium release, all sorts of downstream possibilities. But all of those states of the molecules in downstream events are not necessarily 0 at time 0. There's going to be some basal level of calcium, and it will be in equilibrium with binding partners. There may be some basal level of phosphorylated tyrosine that will be maintained at steady state. So you need to be sure that before you apply a stimulus, that all of the other reactions that are happening at baseline are at steady state. And so that's an important modeling issue that needs to be considered, especially when you're applying a stimulus. Okay, so let's move on to diffusion. This is a nice little cartoon that I like to use that shows that diffusion also can be either thought of as a stochastic process when you have very few molecules, but if you have a large number of molecules, it can be thought of as a deterministic process, a continuous deterministic process that can be solved with differential equations rather than considering probabilities. So in the top, we have a single molecule, and we at the beginning of this little animation, let's let it start again. There's a barrier to the right of this rectangular box, and the barrier opens, and the molecule that's starting at the left of the barrier never, during this short period at least, never gets to the other side. If you have many individual molecules, you can see that some of them do, if they're started off pretty close to the barrier, some of them do manage to get to the other side, but you can intuit just by looking at this animation that how many get to the other side is very probabilistic, and that obviously more molecules will end up on the other side as a function of time. But then, of course, some of those molecules that end up on the other side of the barrier will also, just by probability, go back to the left side of the barrier, where the barrier was after the barrier is open. So how do we solve that? So there are simple, again, probabilities that can be solved using the software involving Brownian dynamics for these stochastic processes involving small numbers. For larger numbers, where you can deal with the concentration regime, then you can solve these in terms of differential equations. And the differential equation that is usually written down for this is just Fick's first law. But the key point is that molecules move down a concentration gradient. And here in Fick's law, you see there's a concentration gradient, the change in the concentration with respect to a spatial variable x, and the flux through that barrier, J, is given by a constant, a diffusion coefficient, times the concentration gradient. Okay. So there's also Fick's second law, and I like to express that also in terms of an equation. Here in Fick's second law, we have the concentration change at a particular point in space as a function of time. And that's given by the second derivative of the concentration gradient. Here I've written it in 3 dimensions. Before I just had 1 dimension. But again, we have that same diffusion coefficient for this molecule I, and the concentration of I as a function of time is given by that diffusion coefficient times the second derivative of the concentration with respect to space. So obviously, right after the barrier is released, that's where you have the biggest concentration gradient, and that's where the concentration will change the fastest at that particular point in time and at the particular point in space right adjacent to where the barrier used to be. Okay? So that's all that this is saying, and it's saying that stuff accumulates if it moves in faster than it moves out. So it's moving into this area where the barrier used to be, and so the concentration increases. But if you're at the left side of the barrier, then of course the concentration would decrease. And this equation describes that. So once again, we can use software to help you solve these equations. All you have to do is tell the software what the geometry of the system is, where the barriers are, where the molecules originate from, and give each molecule a diffusion coefficient, and the software will solve either the stochastic problem, if there are a few number of molecules, or the deterministic problem. Okay? So when you have reactions coupled to diffusion, you have both reactions where you have changes of state of molecules and molecules binding to each other, as well as the molecules diffusing around in space. Then you have to solve what's called a reaction diffusion equation, where you change the solve for the concentration as a function of time of every molecule I by writing down equations like this, or having the software solve them for you. And here R sub I is just the rate at which molecule I changes because of reactions. Okay. So when do we need to consider diffusion? Well, you need to consider diffusion, first of all, if, let's start with the second point here. You need to consider diffusion, obviously, only if the molecules that you're interested in your overall system, are not uniformly distributed at time zero. If everything is uniform, if you have a box and all the molecules are uniform, then no matter what the reactions are, there's never going to be a diffusion gradient, right? But very often in cells, reactions originate at membranes. And so if that's the case, if you have membrane fluxes, or if you have receptor signaling, then the system is not, the molecules are not uniformly distributed because the membrane serves as a heterogeneous region for reactions to take place. Assuming, of course, that the reactions then affect downstream reactions that occur in the cytosol or, say, in the nucleus. So if there are membrane reactions involved, then membrane movement between movement of molecules between membranes and a volume compartment, then diffusion may be important. But then we have the second consideration, which I actually put down here first. Diffusion will only be important if it is limiting, if it limits the motion of the molecules on the timescale of the reaction. So you need to consider diffusion if diffusion is slow compared to the rate of reaction. Okay. And you'll hear more about this, especially when you hear more about spatial modeling later on in the course. But this introductory lecture is just to get you to start thinking about some of these concepts. Okay, so let's talk about virtual cell now. Virtual cell does allow you to solve both for reaction kinetics, symbolized by R sub I, and diffusion, the divergence of the flux. That's the same as that second derivative of the concentration with respect to time that I showed you before. This is just the shorthand way of writing the same thing. So virtual cell does allow you to do that. It allows you to solve for the concentration of every molecule I, or every state of a molecule I in the system as a function of time. And this is just a little simulation from a virtual cell model of a FRAP experiment, where a square region on a membrane is frapped. Hopefully many of you know what FRAP is, and I'll tell you more about it later. You can follow the diffusion of that molecule as it reappears in the bleached area. So that's a membrane FRAP reaction. So we have 26,000 registered users in Virtual Cell. It's been a project that's been going on for about 25 years, maybe even a little less time than Copasi has been around. Copasi has been around for quite a long time as well. It solves reaction, diffusion, advection, and electrophysiology type problems. You can formulate your problems in any combination of those kinds of physics. I didn't mention advection. By advection, we mean flow. If there's a flow of, instead of just diffusion, if there's actually a directed flow of molecules in the system, virtual cell can be used to handle that kind of system as well. And electrophysiology has to do with, of course, changes in membrane voltage and membrane currents, and how those could be coupled to the concentration of molecules that can go through the membrane and enter the cytosol. So we can do electrophysiology as well. The concept of electrical properties of membranes are also covered within virtual cell. By zero-dimensional geometry, I mean a model that just considers the sizes of compartments without worrying about their shape. So zero-dimensional models are, by definition, models that only consider reaction kinetics and transport of molecules across membranes without considering diffusion. So that's what we mean by a purely kinetic system. But you can also formulate your models in virtual cell using one-dimensional, two-dimensional, or 3D geometries that then allow you to also model diffusion. And the geometries can optionally be captured from microscope images, just like the geometry in this simulation that you see at the upper right. And as I described deterministic and stochastic type problems, virtual cell can handle both, either with purely kinetic systems or in spatial geometries where, in the case of a stochastic simulation, you're dealing with Brownian dynamics. In the case of a continuous simulation, you're dealing with the solution of fixed laws. We can do parameter scans where you can automatically set up multiple simulations to check an array of parameters and how that changes the simulation results. So that's what we mean by a parameter scan. And we can also do parameter estimation. The reason we can do parameter estimation is because we are partnering with Copasi. Copasi is superb at doing parameter estimation, as you'll hear later on. It can do it in many, many different ways and solve for much more complex problems than virtual cell. But we've taken some of the Copasi technology and included it in virtual cell so that as a convenience, you can do parameter estimation in virtual cell as well, but in a more limited way. By parameter estimation, just to make sure everybody understands what I mean, is if the typical case is that you have some experimental measurement of some experimental observable, and you want to fit rate constants to that observable in your model. You don't know what the rate constants are, but you know that they have to fit whatever you're observing in your experiment, and so you can use minimization techniques to determine the parameters. Virtual Cell is built on top of a database. So you can store your models on our servers if you wish. You can also search for models that other people have made available on our servers. So that's another component of Virtual Cell, and it's a client-server tool so that you can also run your simulations on our servers. You can run your simulations locally on your own computer and get simulation results, but most of our users find it more convenient to save their models on the database and save their simulations on our database so that they're all collected in a nice way. And then when you publish a paper using our tool, you can make your model public so that anyone can look at it. And I think Michael sent you in one of his emails a link to our website where we maintain links to all of the published papers that have made models public. I'll show you the virtual cell in a minute. Hopefully I'll have enough time to do that. So there are also links to external model and pathway resources that you can access through the software. And these are the kinds of biological problems that virtual cell can handle. Signaling and metabolic pathways, I mentioned those kinds of reaction networks already, intracellular trafficking, ion channels, electrophysiology. And one of the big uses of virtual cell is virtual microscopy. If you do a quantitative microscopy experiment, say on a confocal microscope, where you do something like a photo bleaching experiment like this one, you can analyze that experiment using virtual cell, where you can, for example, in a bleaching experiment, you bleach out the fluorescence from a region and look at how the unbleached molecules in neighboring regions diffuse back into that bleached region, right? But you don't know the diffusion coefficient. So you can use virtual cell to do a parameter scan where you check different diffusion coefficients, and see which one fits the system the best. And for a complicated geometry like this and a diffusion area like that square area that I showed you, there's no way to do that analytically. So using virtual cell to analyze your experiments, and obviously there are many other kinds of experiments, fluorescent indicator experiments, fluorescent sensor experiments, translocation type experiments, that all of which you can do quantitatively on a microscope, but to actually understand what those experiments are telling you, you really need a tool like virtual cell. So a little bit about the virtual cell interface. So the interface is hierarchical, and this is, I always like to give credit to sort of the lead developer of virtual cell, who's been working on the project right from the beginning with a little hiatus, Jim Schaff. And he came up with this hierarchical structure, where at the top of the hierarchy is what we call the physiology. And this is where you describe the reaction kinetics. You describe what all the molecules are, which compartments they're associated with, and what the reaction kinetics for each transformation is. So that's a reaction network. You can also describe this in terms of rules, where you can have a rule-based model, which you'll, again, hear more about in a more advanced lecture by Michael Blinoff. And once you have the reactions and the rate expressions, and which, the identity of the different compartments and membranes that are associated with the system that you're interested in, then you can model those in lots of different ways. So a single physiology can produce many of these components of the model called applications. And you can think of them as different ways of solving the problem depending on what your question is. So at this point, you can do ordinary differential equations. If diffusion is not important, partial differential equations, if diffusion is important, the combination of the Fick's second law and reaction kinetics, partial differential equations. You can do stochastic simulations, non-spatial, again, if diffusion isn't important, but if diffusion and small numbers of molecules are involved, then you can do spatial stochastic simulations. And we've incorporated a standard method for doing that, a standard simulator called Smolden, to be able to do that. And then we also developed a hybrid spatial deterministic stochastic simulator, where you can have some of your species, some of your molecules being simulated as individual molecules stochastically with probabilistic transformations, but others that may be present at very high number that would be impractical to model as individual particles, you can solve those deterministically. So we have that hybrid combination as well. And then there's something else called network-free stochastic simulations, which I'm going to let Michael talk about in, I think, tomorrow, when he talks about rule-based modeling. So these are the different kinds of simulation outputs that you can get. Ordinary differential equation outputs where you have smooth curves, stochastic where there's noise involved and probabilities, a spatial simulation, diffusion and kinetics, a stochastic spatial simulation where you have diffusion and kinetics, but following individual particles. This isn't really a good representation of the hybrid, but it's trying to just make the point that some of the species can be covered deterministically, while others can be modeled, simulated as individual particles. And then this network-free simulator, NFSim, follows the propagation of individual species in a very combinatorially complex system. And again, I'll let Michael talk about that later. So let me make a few more points about the structure of virtual cell. For those of you who are using it, it's very important to understand this. So again, I mentioned the physiology. This is where the biological mechanisms are expressed. And I mentioned applications. This is where you decide what kind of math to use, what kinds of physics to use to simulate your problem. But importantly, it's not just the kind of physics. It's also what specific conditions you want to test. This is where you specify, for example, the initial concentrations of your molecules. And you could decide to try simulations with different initial concentrations. This is where you would decide what geometry to use if it is a spatial simulation. And you might want to try it with different geometries. You might want to try it with an analytical geometry. If you just want to look at sort of a generic cell that's spherical or hemispherical, you can try using a hemisphere as your geometry. But you might want to compare that to what happens in a real geometry that you determine from a stack of confocal images, a 3D confocal image that you get from your microscope looking at your particular cell. So you could make many, many applications that try different ideas, that try different conditions to simulate your system. And so that's why we have many applications that can be created, even if you use the same physics and math for them, but there can still be many applications that can feed off of the same physiology, the same mechanisms. Once you have an application completely specified, there's a one-to-one relationship between an application and the math that's encoded within that application. And Virtual Cell will automatically generate that math, automatically produce the math. And then once you have the math associated with an application, then you can run simulations. And this is where you could try different numerical methods, different durations of the simulation. How long do you want the simulation to run in laboratory time? You can try parameter scans where you try different parameters within the simulation. And so you can have, once again, a one-to-many relationship between an application and its math and the simulations that you generate. So this tool is very, very flexible in allowing you to try very quickly, to try very, very many different ideas as to how your system might be evolving. So you can very readily produce many of these applications, and from it, many of these simulations. So let me go through the VCell user interface a little bit. Again, this will be available in a Google Doc, so you'll see this. If I have a few minutes, I think I will. I'll show you the user interface directly. So this is the biomodel user interface. It consists, first of all, of, in the upper left, what we call the biomodel navigator. So for a particular biomodel, this upper part here represents the physiology, the reaction diagram, the reactions, structures, etc. And below that are the applications. Again, you can have many applications for a single physiology. So this particular model has 4 applications that are labeled. You can name them whatever you want. This pane over here corresponds, in this case, to the reaction diagram. That's what we have chosen in the physiology. And it's the biomodel element. this is the reaction diagram. You can choose one of these others as well. The reaction diagram is the one part of the physiology that is the most useful and that you probably will spend most of your time on. Once you have a particular component of the biomodel chosen, and here, because this is red instead of yellow, this transport icon is chosen. It's been clicked on, and you can see the details of what that icon represents in the lower right panel. So this is where you see the details of the reaction rate for this particular case of what how this would be specified. Okay. And again, we'll play with this shortly. So this is selection details where you produce the rate expression. And then moving over to the lower left, we have the database resources. So this is where you can search for a model either within your own group or one that's been shared with you or one that's been made public. There are also these other database resources in these other tabs that you can access as well. Okay, so now we're moving down to an application, this application called 3D Image, and within that, we've selected the geometry. So this is the geometry associated with this particular application. Doesn't have to be an application that specifies a geometry, but here it is. And here we take each of the structures that were defined in the physiology, the nucleus, the extracellular space, the cytosol, the plasma membrane, and the nuclear membrane, and we associate them with a particular region within our geometry. So that's what these lines are about. And where does the geometry come from? Well, again, this particular one is from a confocal image stack, and it's been brought into Virtual Cell using some tools that we have to segment and define the geometry based on a raw experimental image. But you don't have to do that. You can specify it in terms of analytical geometries like spheres or cylinders, etc. Okay. So I mentioned rule-based modeling. The rule-based modeling physiology looks somewhat different. You can actually combine rule-based modeling with network-based modeling. And again, Michael will tell you more about that tomorrow or the next day. One last thing I wanted to mention before we take a quick look at the software, and that is that you can also directly deal with the math of a model, or you can ignore it. The virtual cell, once you specify a physiology and an application, it automatically produces math, a math description of that system. And here's a screenshot of the math that you can look at. But you can also use this to create a math model. And that produces a completely editable math description in a very simple math description language that you can actually, if you're more comfortable with math, you can actually play with it and edit it yourself. Or you can create it from scratch, because I said the language for doing this is sort of like a MATLAB type language that you can easily learn. Okay, so let's look at VCell. And by the way, we're going to start VCell, but I also want to make sure you all know that there's lots of support on the VCell website. So I'm going to click this and go to the website, and you can see this is the support page, and there are lots of tutorials. There's user groups, there's videos that you can use to teach yourself how to use VCell. But let's escape from here. And I'm going to actually, instead of starting VCell ahead of time, I deliberately decided I was going to start VCell from scratch just to show you one feature of VCell that sometimes can be a little confusing, but actually a very cool feature. And that is that my login credentials are already stored using this automatic login feature going through a Google login on this website. So you can just, after the login, you can just close this, and VCell down here is now open and connected. You can see it's connected over here. That means that it's connected to the database, it's connected to the simulators. You can do a search for, down here in the database area, you can do a search for a model. I'm going to say, look for FRAP. And you can see that under my biomodels, there are 27 FRAP, something that has to do with FRAP. There are 55 that were shared with me. Hopefully you can read this. It's kind of small. And there are 4 tutorials. There are 13 published FRAP models that have something to do with FRAP. Let's see what they look like. You can see these different models. Here's one from 2012 by Ann Cowan, et cetera, et cetera. But let's look at one of the tutorials. Let's look at FRAP binding. So here's a simple reaction diagram that came up. The reaction diagram is highlighted. You can navigate around by just moving to these different components of the physiology. But let's look at the reaction diagram. If I click one of these yellow squares, it brings up the details of that particular component of the reaction diagram down here. If I look at the applications, there's a compartmental application where, going back to the reaction diagram quickly, you see there are 3 compartments that were defined: a nucleus, a nuclear membrane, and a cytosol. And in this compartmental application, they're all mapped to a single point, which means that it's only going to solve kinetics. It's not going to solve for diffusion in this particular case, but you still need to define the sizes of these compartments. So there's a size in terms of micrometer cubed for the nucleus. That's 130.8 something, and 520 for the cytosol. And the nuclear membrane has a surface area. It's units of micrometer squared, and it's 26 micrometers squared. So that's, I don't know where that came from. But then if you look at the spatial model, this is a model, I guess it's of it's a 2D model of a sphere. Well, it's not a sphere, it's just 2D. So it's like a disk where the nucleus is defined as a circle. And this is the inside of the nucleus, and that's the outside. And again, the structure mapping shows how we assign what we call nucleus in the reaction to a nucleus in the image, in the actual geometry. Cytosol is the red area, and the nuclear membrane is the interface between the cytosol and the nucleus. And there may be a simulation here. Yes, I can bring it up just by calling it up from the database. I'm not actually running it now. And then you see the simulation. So this is just a quick overview of how you navigate around a VCell model. So this is the simulation result. And let's look at do I have any time left? Oh, I'm over time. So I'm not going to take any more of Ani's time, and I'll stop here, but I'm happy to take questions in the chat.